and is now approved and used to treat a number of different cancers. The first place where this happened was in a class of medicines called checkpoint inhibitors, or immunotherapy drugs. A lot of people will have heard of these things. PD-1, CTLA-4 are some targets where there are drugs that get infused that hit these things that are on the surface of T cells. And they actually are natural breaks to the T cells. T cells might be in our body there, but turned off or not turned on enough to be strong enough against cancer. And for certain types of cancer, it's been absolutely miraculous that if you make a drug that hits the brake on the T cells go stronger and they can be unleashed against cancer just by taking the brakes off of them.
What sorts of cancers has it been successful for?
The poster child for this has been melanoma. Mm-hmm. One of the big success cases was Jimmy Carter, who had a melanoma, which is a skin cell, aggressive skin cancer that had already gone to his brain, which was thought of as a death sentence. And he got treated with checkpoint inhibitors and basically was cured.
Amazing.
Amazing. And so, you saw these tumors just shrink away, and not just him, but in a large fraction of melanoma patients now respond to these. Mm-hmm. And so that has changed how melanoma is treated. It's in other cancers to varying degrees, because some types of cancers can respond to this. That's taking a drug that unleashes the T cells that are already in our body. The focus of my research the first thing I said was we're living in this amazing moment of biology where we can do things to cells in our body that with incredible precision, and we're often just limited by our imagination. And what we can see now is that we don't actually have to just be limited to the cells, the T cells that are naturally in our body that already have this random distribution of sensors. We can actually genetically make one of these sensors for T cells and put it into T cells. We can put a gene that encodes something on the surface of T cells that will make them programmed to search and destroy for cancer cells. Mm-hmm. Now, this is largely known as chimeric antigen receptor T cells. That's a long term. They're known for short as CAR T cells, chimeric antigen receptor. And what that means, chimeric, is that these are stitched together. This is a receptor that was designed in a lab, does not exist in nature, but can be put into a piece of DNA, delivered into a T cell. And when that DNA goes into the genetic code of the T cell, all of a sudden the T cell will start making proteins that go on its surface and act as these artificial sensors. And those CARs, then when those T cells get reinfused into a patient the way that you get like a blood transfusion, those CARs are directed to go against cancers. This has been done for certain types of leukemia and lymphoma, and there's been these amazing success stories. The thing that woke up me and the world was in 2012, there was a young girl who was the first pediatric patient to be treated with a CAR T cell for cancer. So she's become a heroic figure, Emily Whitehead. She was, I think, 8 at the time, and she had a form of leukemia that it just was, for some reason, whatever reason, it failed all the treatments, and nothing worked. She was going to be sent home on hospice. She had exhausted all the possibilities at the age of 8. And she got enrolled in at that time, highly experimental treatment to get these CAR T cells. So her blood cells were taken out in a big blood donation. Her own T cells were genetically modified. And we can talk about how that was done. It was actually done with like a pretty crude technique that's been around, actually used viruses, lentiviruses. These are sort of modified HIV viruses to deliver this extra piece of DNA that encoded the CAR. And this was done on her cells. And then after that extra gene was put into the T cells were reinfused into her body. And it was not a straightforward course. She ended up in the ICU. The immune system had to, people in real time, people had to figure out how to control the immune system and the side effects. But as that was controlled, all of a sudden her cancer cells disappeared.
Amazing. And the lentivirus itself didn't spark an immune reaction that was
No.
that outweighed the benefits of the cargo.
No, amazingly, it really hasn't. I mean, there's been some discussion about the risks of using these lentiviruses, and we'll talk in a second about how we can do better now.
Yeah,
but
people are going to hear putting viruses into cells and putting them into humans, and a bunch of people will freak out. But I promise you that things like adeno, which is like a cold virus, or lenti, which is similar to HIV, and of course they didn't give her HIV. They changed the virus, so they're not delivering HIV. These viruses are incredible because they can create long-lasting expression of genes that you deliberately put into them. They're a shuttle.
It's an amazing application of biological understanding, right? That all of a sudden we've been studying viruses because of the risk that they have, but we've learned that they can deliver, that viruses have evolved to be very good shuttles and to deliver their genetic material into cells.
The way I think of it is the viruses have evolved to take advantage of our biology and our genes.
Yeah.
And so we did the ultimate touche in these instances. Like, you're so good at hijacking our cells' DNA and proliferating. All right, we'll leverage you to help us as opposed to hurt us, right?
That's exactly right. And so that was done in 2012. Emily Whitehead was 8. It was done as an experimental treatment at the University of Pennsylvania. And the story now is that all these years later, Emily Whitehead is not only cured of her leukemia, she's pre-med at the University of Pennsylvania.
So awesome.
And so
awesome.
No one could ignore that. You know, this was just all of a sudden this dogma that I had just been taught a couple of years early in medical school that we should ignore the cancer immunotherapy. We were just wrong. And all of a sudden, the field woke up and said, okay, the immune system is not just limited to treating viruses and bacteria, or protecting us from viruses and bacteria. The immune system can be exploited and potentially re-engineered to protect us from cancer and maybe other diseases. So that was 2012. 2012 also was the year that a paper got published in Science by Emmanuel Charpentier and Jennifer Doudna that introduced this new technology called CRISPR. And we'll talk about this, but CRISPR fundamentally is a tool to rewrite DNA sequences. That came out in 2012. And on a personal level, 2012 was also the year that I moved to San Francisco to start a lab studying T cells and how genetics influences T cells. I was looking around and trying to figure out what my lab would do. And all of a sudden, I was arriving with an empty lab space at exactly the same moment that the world was shown that T cells could cure cancer, and that we had a tool that could potentially rewrite DNA sequences, and that we wouldn't be limited to these lentiviruses, which are kind of clunky, the best tools we had at the time, but pretty clunky and non-precise in how they insert genetic material. All of a sudden, we could imagine that we could take T cells and use CRISPR to actually pick individual places in the genome and make targeted changes to program exactly how cells behave. And that is the basis for my ongoing work. We've put a lot of work over the years into being able to now take CRISPR technology, get it to work in T cells, to learn the rules about what are the genetic changes that will be most effective at making T cells into immunotherapies that cure patients with different diseases.
Amazing.
And then to go all the way and then actually use CRISPR to make T cells that can be input into patients with new levels of precision and power. And that's in clinical trials now. We're now in clinical trials with these CRISPR-engineered CAR T cells, and we're not just going after leukemias, where these CAR T cells have historically worked, but we're also thinking about, can we make these work for the really common causes of cancer deaths, solid tumors? And that's been a challenge, and we can talk about that. But getting T cells to find the right targets in tumors and then work inside of tumor environments, which are inherently immunosuppressive, requires figuring out additional gene edits that are now possible with CRISPR to try to beat the cancer at its own game. If cancer is evolving to make itself cloaked from the immune system, now with CRISPR we can think about getting one step ahead and making T cells that are able to resist all the tricks that cancers throw at it to be more, and I think we're on the brink of having precise CRISPR-engineered cells that will, I hope, start to melt away cancers without the side effects of chemotherapy.